Students, let us continue with sphagnum, the sporophytic generation of sphagnum. Now, before the development of sporogonium, what are the post-fertilization taking place in the sporophyte? The zygote begins to enlarge in size and secretes a wall immediately after fertilization. The early development of embryo takes place in succession of the transverse walls to form a 6 to 12 celled filamentous embryo. Now, the upper cells of the filamentous embryo divide by two successive vertical walls, each cell forming a quadrant. The quadrants divide by periclinal walls and separate the peripheral amphitheecium and the central endothecium zone. The cells of the endothecium rapidly divide and form a mass of sterile tissues with a rounded apex, the columella. Further, a periclinal division occurs in the amphitheecium cells, delimits the inner fertile layer, the archisporium, from the outer sterile tissue, the outer amphitheecium or the capsule wall. The 2 to 4 cells thick archisporium forms a dome-shaped sporogenous mass of cells. Each sporogenous cell acts like a spore mother cell and divides by meiotic divisions to form 4 haploid spores. The spores, they develop into the new plant by the germination of the spores. The whole sporogonium develops enclosed within the archegonial wall. The archegonial wall forms the calyptra. At this stage, the archegonial neck appears, and with the maturity of the sporogonium, the sporogonium it pierces out through the calyptra and above the perichaetal leaves due to the elongation of the female gametophore, that is the pseudopodium, which is the false foot, and the terminal portion of archegonial branch swells due to the bulbous foot. It is called the vaginula. And the details you can see here, the development of the sporogonium. Figure A to N shows the stages in the development of the sporogonium, and you can see the zygote after the fusion of male and female gametes. It divides by two divisions to form a epibasal cell and a hypobasal cell, and the filamentous structure is formed, and the lower cell gives rise to the haustorium, and the periclinal division gives rise to the amphitheecium, which is outer, and the endothecium, which is the inner layer. And figure O, you can see the mature sporophyte or the sporogonium of sphagnum, having the neck, calyptra, operculum, annulus, the spore sac, which is horseshoe shaped in structure, the surrounding by the jacket layer, and the inner is the columella, and the outermost layer of the sporogonium is called the epidermis, and it is the sporogonium consists of the foot and a pseudopodium. And so this is the development of the sporogonium in sphagnum. Now, what about the germination of the spore? Once the spores are formed, the operculum gets off as a lid, escaping forcibly yellow powdery mass of spores. Before coming to the germination, let us discuss the structure of the sporogonium. You can look to the figure O. I have explained before. See, the structure of this sporogonium consists, the body of this sporogonium consists of a large bulbous foot, a neckline inconspicuous seta, and a spherical capsule. The capsule wall is several layered thick. The outer layer is heavily cuticularized, and the epidermis forms a ring-like annulus towards the apical region of the capsule, delimiting the disc-shaped operculum from the capsule body. The capsule has a central columella overarched by a dome-shaped spore sac that contains haploid spores. The whole sporogonium is shifted out of the perichaetum due to the prolongation of the archegonial branch, that is the pseudopodium. The solen sac-like distal end of the pseudopodium encloses the foot of the sporogonium, and which is called the vaginum. So once the spores they get dispersed out, the spores they come out in a powdery mass, and once they come out, the spores they germinate to form the spores. Now, how does the germination of spores takes place? And the spores germinate to form the protonema, which is the spore. Once the spore is formed, the plant is shifted towards the gametophytic generation, the young gametophyte. The spore are tetrahedral with a prominent triradiate mark, and the spores may or may not germinate immediately after dehiscence. They can remain viable for a period of 6 months, and during germination the exine ruptures along the triradiate mark, and intine comes out in the form of a germ tube. B. The germ tube or germ papilla divides by transverse walls and forms a 2 to 4 cell filament or the protonema. You can see the figure D. Soon the apical cell of the filament divides by 2 vertical walls and forms an apical cell with 2 cutting faces. Figure F. Each cell contains abundant chloroplast. The apical cell divides to form thalloid, one cell thick primary protonema. Figure G you can see. Here you can see the protonema. Soon the activity of the apical cell stops and the marginal cells become active to form the irregularly lobed thallus. Figure H you can see. And sometimes the marginal cells form the separate filament that develops into a new flat thallose secondary protonema. So primary protonema से secondary protonema भी बन सकता है, जरूरी नहीं है लेकिन it can be formed. The multicellular obliquely separate rhizoids develop from the basal part of the protonema. Towards the basal portion of the thallus, a tetrahedral apical cell with 3 cutting faces develops and forms the segment, which in turn gives rise to the stem and leaves, that is the gametophore. Each thallose protonema gives rise to a single gametophore. The juvenile leaves consist of uniform hexagonal cells, but the cells of the subsequent leaves differentiate into chlorophyllose and hyaline cells. You have seen the leaf structure of the sphagnum. Now we have discussed the economic importance of sphagnum, affinities, the gametophytic generation and the sporophytic generation. Finally, to bind up, we come up to the alternation of generations in sphagnum. So, this is the diagrammatic representation of the life cycle of Sphagnum. First, we begin with the gametophytic generation, which is the N generation. I have told you the spore tetrad, the spores, they get separated from the spore tetrads, and the protonema is formed, and the main gametophore develops. And when the plant matures, the gametophyte matures, it bears the antheridial branches and the archegonial branches. The plants may be monoecious or they may be dioecious, and the antheridial branch gives rise to the antheridium, and the archegonial branch gives rise to the egg. Both fuse to form the zygote, and zygote, it develops into the embryo. The lower portion gets turned into the hostorium, and finally the sporogonium is formed, and the spores, they remain in the spore sac, and which on undergoing meiotic division gives rise to the spore. Archisporal tissues gives rise to the spore mother cells, and the spore mother cells finally undergoing reduction division gives rise to the spores and gives an end to the sporophytic generation. So this is the diagrammatic representation of the genus Sphagnum. With this, we finish with the order Sphagnales. Thank you.